Yes, Vidyashree, kindly mute yourself. Can you mute yourself? You're unmute currently. Yeah, good evening. Can you mute yourself? Like there is a background sound in your. Yes, thank you. Okay, I think like people are still joining in. Like it's 6:04 now. Maybe we'll start in a minute or so. There are around I think 13 of you all. Let's see if anyone else joins in, and then we'll start. Yeah. I think I should change my host control so that everyone can join. Okay. So as this is the first lecture. I will. This is majorly would be an introductory lecture of the whole course, and we'll discuss more about it in the coming time. Okay, can you all see my screen? Give a thumbs up, please. Yeah, thank you. Or else you all can write it in the chat box, or you all can unmute yourself and like speak as well, whichever is comfortable for you all. Can you see the presentation as well? And if I make a full screen, oh, I can still see this. That's great. Okay, I think we can start. It's around 6 already. I think most of whoever wanted to join might have joined, and whoever might be a little late, they might be joining soon. Okay, so good evening everyone. I'm myself Gaurav, and I am the TA for the course called as Bioengineering and Interface with Biology and Medicine. This is a course of NPTEL which has a code of NOC 26 BT13, and this specific course is taken by Professor Sanjeeva Srivastava. So you all can go through his lectures as well. Like in the first week, there are 5 lectures which Sir has taught, and we are the one who actually help you all in order to understand if you all people are having any kind of doubts after going through those 5 lectures. So I don't know if you all have went through those 5 lectures. Anyone who have went through those 5 lectures, or just looked around it, like if not completely. Yes, anyone has already gone through those lectures by Professor Sanjay Srivastava? If not, then you all can obviously say no as well. There's no harm in that. Okay, so I don't know if you all know that, like so you all might be like if you all go through your course, you all might end up saying something like this. So in this week 0, there was an assignment. If you all need to solve that, and then in week 1, you all can see these 5 lectures here. You all can click on each lecture like this, and I'll just show you so that you all can go through that as well. So, like, there are certain lectures already taken by Professor Sanjeeva in order to uh, explain you all about the course itself. So, in this week one, sir has gone through these five specific topics, and there is a specific assignment also given to you all. I think the end date is around 2nd Feb or something like that. So, you all can go try and solve this assignment also and try to so basically, what we do as a TAs in terms of NPTEL courses, so you all go through those lectures. Oh, okay. Ananya has already went through those lectures. Great. I think most of you all have not gone through those lectures. Even that is fine, but you all need to go through those lectures so that after coming here, it is easier for you all to ask certain questions related to the course itself, like whatever sir has taught. For example, if you might have not got what sir has exactly taught in between, or you might are little confused between what exactly sir was talking about, then we are here to explain you, like, clear those doubts basically. And along with that, what I'll be doing is, uh, along with those course, I'll give you these course details like after some time. So basically, what I'll be doing in this specific lecture is whatever sir has gone through in this week one lectures. So in week one, sir has took five lectures, and in that five lectures also, sir majorly explained about the in lecture one, sir has talked why biology is required for engineers, and in lecture two, like one and two are basically motivational lectures, like why engineers should also learn biology and why biologists like basically how engineers can help biologists in terms of solving a number of different issues. So these two lectures were basically about motivating you all in order to take biology, sorry, in order to learn biology if you all are doing engineering. And like from lecture three, sir has started about the fundamentals of the biology, like what is a cell, basically how life processes happen, etc. Like in detail about how exactly cell replicates, or what all components are present in the cell, and how exactly we the cell survives basically. All those like the cell components, etc. How communication between cell to cell happens. All those concepts were explained in lecture 3 and 4. And in the lecture 5th, there was a talk by one of the doctor who talked about neurosurgery and how exactly uh, the neurosurgeons work, and how exactly engineers have actually helped neurosurgeons in order to treat those diseases with the like, for example, if they want to remove a tumor, like what all kind of machines were developed, and how exactly they are helping them in order to treat the disease. So all those I'll be trying to cover in this lecture as well. So we have 2 hours with us. So what I'll be doing is, in the initial hour, or maybe 1 hour 15 to 20 minutes approximately, I'll try to recap whole of these five lectures for you all, so that whatever you might have already gone through, or you might have not gone through it, whatever might be the case, so that you get revised of whatever things sir might have taught to you all. And that's what I'll be going through in the initial part of the lecture, and later we'll discuss certain question and answers, whatever were the previous year assignment questions, so that it becomes easier for you all to solve the assignments currently given to you all. And coming to the background of the whole course, this is bioengineering and interface with biology and medicine. This is a 8-week course on NPTEL, and the course runs from 19th Jan to March 13th. And this is your first TA lecture. Your assignment zero might have been introduced to you all or given to you all in during Jan 19 or 20, something like that. So, in terms of this course, you will be given weekly course material as well as assignment. And the weekly TA sessions will be on Tuesday itself on 6 p.m. to 8, from 6 p.m. to 8 p.m. And I am the one who will be taking those sessions. And myself, Gaurav, and I am from IIT Bombay itself. And I have been allotted this specific course for in NPTEL to solve your doubts in terms of whatever you people might be having. And the exam date for this specific course is on March 29th. And I think the registration deadline has already been closed. It was March 20, sorry, Jan 26 or 27. So whoever have already registered, like you all can attempt your exam on March 29th. Before that, in this specific course period, I'll be taking 8 lectures of those 8 week course, and we'll try and discuss everything which whatever possible for us. Okay, so till now, I hope you all got what I'll be doing in terms of TA lectures. I haven't completed the whole first week lectures, but I did go through first week lectures. Okay, the exam is online mode. Yes, probably online mode. I can confirm it by asking to the NPTEL people. It will be most probably online mode itself, because I think people are registering from whole India and from like every corner of the India, so though we don't know exactly, so we cannot have a in-person exam for this course. What is the exam form deadline? I I think for exam form you all can apply later also, but for registration, Jan 26 was the last, or Jan 27th was the last. Yes. So before that, like before starting the course, I wanted to know like your background as well. So you all belong to what kind of background, so that accordingly I can discuss certain topics in detail or superficially. If you are already from a biological background and if you all know certain stuffs, maybe I can go through it a little faster. But if you all are from a different background, like if any one of you, like maybe currently we are 21 of us, I think 25 of us. So if some of you are from a very different background, then I might need to go through specific parts of the lecture in a bit more detail, so that even they can understand. So people are from BSc Biotech, Biotech, BTech Biotech, MSc Biotech. Great, like you all know basics of biology. Then how about others? BTech Biotech, fourth sem BTech Biotech. Okay, most of you all are from BTech Biotech itself. BTech Bioengineering. Great, great. Okay, most of you all are from a biotech background itself. Anyone out of biotech and doing a different type of engineering? BTech CSE, medical electronics. Oh, great. What does CSE stands for? Like, if you can say that as well. Computer science engineering. Yeah, biotech genetic engineering. Great, biotech. Oh, there is someone from bioinformatics as well. Yes, Anu, do you want to say something? You have raised your hand. Biomedical engineering. Okay, I think most of you all are from a biotech background. Only one person is from computer science background. Anyone else who wants to write the course which you are currently pursuing, so that accordingly I can talk in the lecture, like in detail or superficially, depending upon the topic. Yes. Okay. I think most of you all are from biotech background itself, but few are from initial period, like in the BSc first semester itself, and few are from later period, maybe fourth sem or something like that. So the fourth semester people might understand much more easily as compared to the first sem people. Similarly, uh, the one who is doing biomedical or medical electronics, you people might uh, need a bit more understanding. Similarly, computer science engineering. So Luther, like I'll be going through the lectures. If you have any kind of doubts and if you want me to talk in bit more detail regarding that specific topic, write it in the chat box or unmute yourself and speak to me so that I can accordingly explain in bit more detail if you are not understanding it. Okay. Okay, so then I think we can start with the lecture now. If I already know the background, what all you are from. Just a minute. Why is this? I actually wanted that specific screen on the sides here. Okay, let it be then. I think it's not coming. Okay, so this is the so as I said, I'll be initially going through the recap of all whole 5 lectures, and then talking about then we'll be discussing certain question and answers so that it will be easier for you all to solve the assignments. Okay. So what Sir did in the initial 2 lectures is he went through uh, give certain motivation for engineers in order to pursue biology as well, or like learn biological concepts too. So initially Sir discussed about the global problems present in the world and how biology is trying to solve those issues and how engineers can help us to solve those issues. The first specific problem Sir talked about is the global problem, which is world population. So there is increasing world population, and for that increasing world population, we need to have that much amount of food as well. There is no increase in land as such currently, but if you see the population is being increasing very tremendously, and it is expected to grow till 9.3 billion by 2050. So science needs to be more. I'll just on my pointer as well so that it is not bad. Okay, so there are different factors which can help in increasing the food production, but those specific factors, there are a number of different factors which can limit the food production, such as abiotic stress. Maybe then in certain regions there is very high amount of light, in certain regions the temperature is very high, in certain regions the water is not that clear, like there is saline water. So there are a number of abiotic stress in certain regions which limits on food production worldwide. And there is only 20% of the agricultural land in the world under salinity stress. So this specific agricultural land will not be able to produce certain crops, whereas certain crops can be produced which can tolerate salinity. So these were certain examples basically, like what are the environmental impact on food production. So how exactly we can solve if these current problems exist, like we need to come up with uh, certain strategies in order to eliminate these specific problems and still able to grow the uh, crops which we want for our food, because the current world population is increasing tremendously. So one of the way of solving that specific issue is recombinant DNA technology. So in terms of this, what happens is there is a plasmid which is an extra chromosomal DNA, and we whatever is the gene of interest. For example, if we want to increase the uh, salinity power of a specific crop, or if you want to increase the uh, ability for the crop to sustain high temperatures, or ability of the crop to sustain pest control, like if it will not be dying if there are certain insects attacking that specific crop. So what happens in terms of that concept is the crop will be producing certain stuffs. So because of that specific uh, ingredients which that plant is producing, if a insect comes and if a pest come and attack that specific plant, it will, by consuming those specific products, the insect will die itself and the plant will be surviving. So that can be one of the strategy. But for making that strategy or making that plant to make that product, or or to increase the ability of a certain crop, we need to engineer that specific crop or a plant. So for engineering that, whatever is the gene of interest which has that specific ability, we'll try and insert that specific fragment in the plasmid itself. By it's nothing but a copy paste thing. So you need to cut, copy and paste thing. So you just need to cut this, you need to copy this specific thing and again paste it. It's in a layman language, that's how it is. Like, for doing this, there are certain restriction sites available. These restriction sites are the one which help you all to cut and paste accurately. So they are the one which help in increasing the accuracy basically. And that's how the whole plasmid can be made with the gene of interest which we want. And for joining this, for example, if we want to join anything, we need glue or something like that. So here the DNA ligase is the one which is acting something like a glue thing, wherein the gene of interest, which was not initially present in the plasmid, but we are adding that specific gene of interest into the plasmid, and by a process called as restriction digestion ligation in terms of biological language. But in terms of layman language, you can just say that cut, copy and paste. And after doing that, this is how the plasmid is made. And so the other thing is, we want to deliver this into the organism so that it makes those specific proteins. Basically, it is so if you might have heard about the central dogma thing, the DNA is converted to RNA, RNA is converted to uh, the protein itself. So ultimately, this gene of interest will make certain protein, which will do its function of either, depending upon what gene you are introducing, it can either uh, increase the ability to tolerate high temperature, high salt, or can tolerate certain pests, etc. So all this depending upon the type of gene you are inserting. So there are different ways. One of the ways called as transformation. There is something called as transduction. There is something called as conjugation. There are different ways to insert that. So these were certain examples basically, like the pest resistance. So depending upon the gene, you can make the plant to resist the pest as well. You can also use to alter bacteria for cleaning up the toxic waste. You can increase the growth hormone by accordingly inserting that those specific genes into the body, or else you can block, basically dissolve blood clots by accordingly inserting those genes into the body. So one of the example is streptokinase, which helps you all to uh, dissolve blood clots. So that helps in curing the uh, heart diseases, etc. So how exactly this plasmid is all about? Like, what is this specific thing which is carrying the gene of interest? So this is how it. This is one of the example of one of a plasmid which is PBR322, and this is how a plasmid looks like. It also has a antibiotic resistance gene. So antibiotic resistance genes are basically there in order, which will help you all for a selection process. For example, there is an organism which is not being engineered, and there is an organism which is being engineered. How will you differentiate between them, whether that is engineered or not? So one of the way of selecting the organism from the wild type one as compared to that of engineered one is checking for the antibiotic pressure. And if we just because this plasmid is being sent to an organism, and if an organism is engineered, that will tolerate that antibiotic pressure, and whereas wild type will not tolerate because it is not having that gene to resist that specific antibiotic. So that's so this antibiotic resistant genes are present for in order to help you all for selection process. Similarly, there is these are called as restriction sites, and these are the one which will help you all to engineer basically to cut and paste stuff. So your gene of interest will also have this specific restriction sites, and this can be cut accordingly, and accordingly your gene of interest will also be digested, and they can be joined together with the help of DNA ligase. This is one more example of one of the other vectors. So there are a number of different vectors available. And in terms of cloning things, so for example, these were the fragments which were gene of interest which is there, and you want to insert it into the phage. So this specific process is called as transduction, wherein you are using a virus for transferring the plasmid or a gene of interest into an organism. For example, this specific phage is being inserted into the bacteria, and it that specific phage has sent that specific fragment, these specific fragments, into the bacteria, and then that bacteria is multiplying that specific gene of interest. Are you all getting it? This specific process is called as transduction, and there's something called as transformation and conjugation also, wherein after using restriction digestion and ligation method, the plasmid is made, and the plasmid is transformed into the bacterial system. Like this, it goes inside the bacteria, gets integrated into the genome, and that is called as transformation. And a process called as transduction, wherein one of the strain is already engineered, and because of the help of conjugation, like the other organism which you want to engineer, they join together, and the DNA which is needs to be transformed can get transformed into the other strain, and even this strain can get engineered. So there are three different ways in order to send that specific this specific plasmid into an organism. This process is called as transduction. This process is called as transformation, and this process is called as conjugation. So these are three different ways in order to send this specific plasmid into an organism for engineering and making this specific organism to express the gene of interest which you want. Are you all getting it? And do I need to go a little quicker if you all know all about all of these, or this speed is enough? Yes, I think I can go a bit quicker as well, but I want your confirmation. Yes, people. This is fine, or do you want me to go a little faster or a little slower if you are not understanding it properly? Because we have only 2 hours and we also need to go through question and answers as well. We are familiar with most of these topics, so you can go a bit faster. Okay. Is everyone with Khushbu? Okay, then I'll go a little faster then. How about Luther, as you are from a different field? Okay. Okay, I think I can go a bit faster, so I'll go a bit faster now. Okay. So that was one of the problem which sir discussed in terms of different problems for crop for agriculture basically as the population is increasing. Then sir discussed about other problems as well, like other global problems. There is climate change happening. There is seawater level somewhere increasing, somewhere going down. There is in some regions there are glaciers which are melting. In some regions there is because of the temperature the land is being not utilized properly. There are a number of different climatic changes and a number of different problems being observed. Similarly, in terms of human diseases, may it be AIDS, cancer, tuberculosis, there is rise. Whatever different types of diseases they are, even they are tremendously increasing, and the way our medical uh, strategies in order to eliminate these diseases, even that is going at a good pace, but not at the same pace at the level which diseases are increasing. So there is a greater need of an engineers to and biologists to integrate and come up with a better strategy so that we can eliminate such diseases or eliminate such problems from the world. One of the way by energy resources have been made better in terms of when an engineers and biologists came together in terms of biofuel, in terms of wind power, in terms of photovoltaic and geothermal. These are different alternative energy sources which were possible because of integration between biologists and engineers. So these are certain reasons why biology for engineers. So there are major life problems require interdisciplinary skills for effective solutions. And similarly, genetic engineering and biology aims to provide promising solutions. Whatever problems we did discuss right now, so biology has a capability to to solve those issues, but biologists can do much better if engineers are with those biologists, like for trying to detect something, for making certain machines or whatever. If engineers are there, biologists, it becomes much more easier for biologists to solve those issues. So this was the lecture one, what sir talked about, and in lecture two, sir discussed about how exactly certain engineers have used biological concepts in order to get success in terms of their engineering thing, which is called as bio-inspired engineering, wherein you look at the nature and you look at certain elements which and from which you take inspiration, and after you take that inspiration you try to solve or your like other human problems or different problems by developing certain designs or models depending upon the nature which you looked at. So these were certain examples which sir talked about. One of the example was bio-inspired engineering for architecture. So you might have seen termite molds in general which termites build, but if you look at these molds, the internal temperature is generally maintained or cool in nature. So in one of the building which is in the Zimbabwe, which is called as East Gate building, they have utilized similar concept like this, and they have tried to make self-cooling system inside the building itself. So this was something which was biologically inspired, and that's what engineers have used and tried to make it much more economical in terms of keeping the internal environment of building inside the building cooler. One of the example in terms of transportation, the Japan people, in order to make their bullet train, they were inspired by the kingfisher. So if you have ever seen kingfisher, when it tries to attack inside the water, if when it tries to catch fish, so it goes at a high speed, and uh, in the water itself, the beak goes in such a high speed that it can catch the fish immediately as much as possible and have its specific dinner or lunch, whatever. So inspired by this specific shape of beak, they have been able to build much more faster bullet trains, which were inspired by the kingfisher. Similarly, there are certain mega projects, for example omics study, wherein which helps you all to if you might have heard about omics, which is metabolomics, proteomics, or so in terms of all of these omics and system biology study, there is a genome which is called as your DNA. If you are studying that specific part of the whole body, which is called as genomics, and DNA gets converted to RNA. If you want to study that RNA, that's called as transcriptomics. It gets converted to protein. If you want to study about protein, it's called proteomics. Then whatever metabolites like carbohydrates, lipids, etc. If you want to study about different cycles happening inside the body, that's called as metabolomics. And pheromones, then it's called as phenomonomics as well. So in terms of this specific different mega projects, if you are this basically omics profiling or omics study helps you all to get a personalized medicine as well. So currently in foreign countries, people are going through different omics study in order to build personalized medicines, and these are very huge projects, and such complex is our pathway inside the body, and these are very much huge projects in order to come up with certain conclusions out of it. So there are certain omics studies which are currently going on, and in terms of IIT, people are trying to make a mobile based portable diagnostic system which are urine analysis or blood sugar test analysis. So in terms of all of these, like what sir tried to say is, so in order to build this specific machine, we need a help of engineers. But how exactly you need to build that machine? What all parameters should this machine should detect? Only biologist will know. Like a biologist understands the biological concept much better as compared to engineers. But in order to make these machines, engineers are the one who understand better than biologists. So that's the reason integration of engineers and biologists is required. Similarly, there are certain mobile phone based diagnostic platforms for detecting sickle cells in blood as well at point of care. So even if you just add a drop of blood into the system, the microscope, like with the help of mobile and with the help of certain slides and the microscope, pick nature of this specific machine, you all can see whether those blood cells are having in sickle shape or not, and that's how you all can detect the whether a person is suffering from sickle cell anemia or not. So these were few examples which sir talked about, and similarly, in terms of life processes in lecture 3, what sir went through is the energy process, the order of sunflower, all this. So how exactly blood regulations happen, and in terms of how exactly this Venus flytrap responds to the environment. So these are different life processes happening at different organism level. So how exactly this systems work in terms of organizations? There are so there is a hierarchy basically, and that's what hierarchies are talked about in terms of these specific examples. In terms of organization, so there are levels of biological organizations. Initially, there is biosphere ecosystem. That in that biosphere there are certain ecosystem. In that ecosystem, there are certain communities. Uh, in a community there are different populations are made up of different organisms, and an organism is made up of different organs, and in that organ itself, like each specific organ are made up of certain tissues, and each tissues are made up of different cell organelles, etc. So that's what an hierarchy system is in terms of biology. And in terms of that itself, what is something which is actually governing each and everything is your DNA itself, which is called as genetic information, or which gives genetic information, and which is also called a central dogma of our life. And so basically, sir talked about different factors which belongs to life. That is something called as evolution, something called as interaction, and something called as energy and matter. Similarly, so I talked about certain organism level thing stuffs as well, how distinct we are in terms of morphology, and how remarkably the biochemical changes happens inside a body, like how different a fruit fly is than a bacteria and than a Arabidopsis thaliana or a human organism. Although such different phenotypically, internally everything is controlled by this specific DNA molecule itself, and few of the enzymes or few of the genes are exactly similar to that of what is present might be present in a bacteria, or something in the plant which is very similar to that of something which can do photosynthesis, etc. So so what sir tried to talk about in terms of all of this is, this is the one which is controlling everything, which is called as DNA. And like where this specific DNA is present inside inside an organism itself, wherein in the organism there is something called a cell, and in the cell the DNA is present. So the sir talked about different components present inside the cell, and how complexly that specific cell functions. And in order to help engineers to understand how that specific cell functions, sir has given an example of a car itself. So there are different components present inside a car. Imagine this car as one cell. So in that one cell, there are n number of different components present, and that's how a car functions. Similarly, in terms of a cell, there are n number of different components present. Only if they function simultaneously with each other, in accordance with each other, then only a cell will be able to function. That's what sir wanted to say in terms of this. And how small a cell is, this is in terms of in 100 micrometer range. And just to give you uh, how small a cell is, like most cells are in between 1 to 100 micrometer in diameter. So generally bacteria are much more smaller than the cell of an eukaryotic organism. So there are different microscopes which have been built by the engineers in order to help biologists to study cells which are microscopic in nature. So there are different types of microscopes which are being made, like bright field. There is phase contrast microscope. There is SEM. There is STEM, scanning electron microscope, transmission electron microscope, etc. Every microscope has its own property to study. Like bright field helps you all to study if you are staining the cells. It will help to differentiate between uh, depending upon the type of stain you are using. Scanning one electron microscope tells you about the upper layer of the cell, the superficial nature of the cell, whereas transmission actually goes inside the cell and talks about the so as the name suggests, it transmits the cell itself and tells you about the internal structures of the cell or whatever you want to look at it. So in terms of cell itself, very initial cell was prokaryotic cell. So prokaryo indicates that there was no nucleus. Karyo means nucleus. So you can see is karyon means nucleus. Pro indicating before nucleus. So these prokaryotes are the one which doesn't have nucleus, and eukaryotes are the one which have nucleus. This is how the cell looks in terms of a eukaryotic cell. This is how an animal cell looks. This is how a plant cell looks, and these are certain components present inside the cell. Similarly, in terms of classification of those organisms, this is how they divided they are in terms of eukaryotes, bacteria, and as well as archaea. Initially, archaea was a part of bacteria itself, but now they are divided depending upon their different pathways which are involved in terms of these organisms, which now are called as archaea. So archaeas are more similar to eukaryotes, but they are still different. And initially, as I said, they were a part of bacteria like archaea. However, they're more similar to eukaryotes than bacteria. That's the reason they were given a different name itself, which are now called as archaebacteria. So this was an animation video. If you are going through those lectures, so basically biochemical uniformity of an organism, which is nothing but the DNA itself or the metabolic pathways, no matter how they exactly look externally or whatever these organisms are internally, some of the pathways are very similar to each other, and ultimately everyone are controlled by a unit which is called as DNA. Similarly, sir explain in much more detail about how exactly these organelles are. This is mitochondria, this is Golgi apparatus, this is nucleus, then there is a cell membrane, and then there's something called a cytoskeleton, which helps you all to. Uh, so cytoskeleton indicating cell skeleton, which makes the cell, gives the cell its shape. Similarly, in terms of plant cell structure, there is chloroplast, which is absent in animal cell. Chloroplast is the one which helps the plant to do its photosynthesis. Then in this topics, after explaining about the cell, sir explained about each and every cell components. Like this is plasma membrane, which helps you all functional separation between cell to cell, and it helps for molecule transport. Similarly, inside the cell there is cytoskeleton. These are cytoskeletons which gives the structure to the cell, which are microfilaments, microtubules, etc., which are cytoskeleton. Then there is nucleus, which incorporates the DNA, the main information of the whole cell, contains genetic information. Then there is endoplasmic reticulum, which helps in protein production itself, processing and transport of protein. And they are divided into two parts: rough endoplasmic reticulum and smooth endoplasmic reticulum. Rough one has ribosomes with it, which helps in protein synthesis, and it doesn't have any ribosome. So it helps in packaging of proteins. The smooth one. Then an organelle called as Golgi apparatus, which is helps in protein transport basically, and also exocytosis in terms of if it wants to excrete outside the cell or keep it inside the cell. Golgi is the one which basically does the post translation modification, and accordingly whatever is needed with that specific protein is proceeded after it goes through Golgi vessels. Peroxisomes are the one which helps in which contain oxidative enzymes, and that helps in degrading peroxide, etc. It also helps in breakdown of fatty acid molecules. Then there is mitochondria, which is the powerhouse of the cell, which has helps in ATP synthesis, NADH synthesis, etc. So these were certain components present inside the cell, which are talked about. In terms of evolution of cells, there is endosymbiotic relation happened. Like initial organism, the prokaryotes have mitochondria with them. Then there are certain prokaryotes which were able to do photosynthesis, which had chloroplast in them. Depending upon these specific when this engulfment happened in terms of endosymbiotic relation happened, so whatever mitochondria is present in the eukaryotic cell, they are being said that, so once this goes inside, so this outer membrane is a part of the eukaryotic cell itself, whereas the internal membrane of the mitochondria is a part of prokaryotic cell. That's how. So if you can see here, like when it was getting engulfed, this outer layer is a part of this specific cell itself, which is an eukaryotic cell, whereas this specific part is a part of bacterial genome itself, and the whole bacteria itself, which, so that specific membrane is coming from bacteria, whether the outer membrane is coming from the eukaryotic cell itself. So if you all have read about mitochondria or chloroplast, they are double membrane structures, wherein the outer membrane is from the eukaryotic origin and the inner membrane is from a prokaryotic origin. So also explain certain differences between prokaryotic and eukaryotic. Like generally prokaryotes are much smaller, they do not have nucleus, whereas eukaryotic has that. And they are larger in size as well. In terms of the amount of DNA, it is multiple linear DNA molecules are present, whereas in terms of prokaryote, it is single cellular circular DNA. Number of base pairs are also lesser in prokaryotic as compared to that of eukaryotics, and the transcription translation process is a bit similar, but this doesn't have nucleus, so it happens everything in the cytoplasm, whereas in terms of eukaryotics, transcription happens inside the nucleus, translation happens outside the nucleus. So in terms of present and absent in the eukaryotic itself, we can divide them into plant cell and animal cell. So generally plant cell has a cell wall, whereas animal cell doesn't have a cell wall. It also has vacuoles, it has plastids. In terms of plant cell, animal cell doesn't have plastids, even it doesn't have glyphosomes as well, whereas lysosome and centrosome are present in animal cell only and not in plant cell. Then so discussed about how exactly membranes are made in terms of gram positive and gram negative bacteria. This is about prokaryotic itself. Gram positive bacteria have a thicker peptidoglycan layer as compared to that of gram negative bacteria, but it also has one of the membrane called as outer membrane. Basically, it is double layered membrane. There is plasma membrane and outer membrane. Whereas in terms of gram positive bacteria, it doesn't have any outer membrane, but it has a greater protection with the peptidoglycan layer as compared to that of gram negative bacteria. And inside the prokaryotic itself, the ribosomes are made up of 23S RNA and 50S RNA, and 16S and 30S are present. So sorry, these are present in the prokaryotes, whereas these are present in the eukaryotes. The ribosomal nature in prokaryotes and eukaryotes is different, and in terms generally the ribosomes are divided into larger subunit and smaller subunit. The ATS ribosomes are present in eukaryotes, and they are divided. This specific part is divided into parts something like that. ATS, which has 28S, 5.8S, and 5S. So these are the subunits of the ribosome itself, and whatever the smaller values you are seeing, 5.8S, 5S, and 20S, those are mRNAs present inside the ribosome. Then this is the same thing which we just talked about in terms of endoplasmic reticulum, rough one, smooth one, etc., and their functions. So this is the whole transcription process. Once the protein is made, they can be transported with the help of Golgi apparatus, which we just talked about. And if it wants to be secreted outside, accordingly, those specific signals will be attached in the Golgi apparatus and it will be taken out. then there is nucleus. Then how does cell communication happens? So that happens with the help of certain cell communication or cell signaling factors. In terms of plasma, in terms of plant cell, the communication between these two cells is happening with the help of something called as plasmodesmata. Whereas in terms of animal cells, it happens with the help of gap junctions. Similarly, there are other ways of communicating as well. One of the ways called as neurotransmitters, wherein in the nerve signals itself, there are certain neurotransmitters which helps in to which sends the certain signals accordingly. Similarly, uh, there's something called as paracrine signaling, autocrine signaling. So this is something called as paracrine signaling, whereas one cell is communicating with other cells accordingly. This we just discussed bacteria, archaea, and eukaryotes. And other way of similar to that of neurotransmitters, there's some communication can happen with the help of hormones as well. And this is something called as long distance communication. Like one of the organism makes the hormone, and it is with the help of the bloodstream itself. It goes through the blood vessels and it reaches the target organ, and that's how communication happens in terms of hormone signaling, which is also called as endocrine signaling. And in terms of plants, also there are certain signaling factors which, like in terms of hormones itself, which helps in regulation of plant growth. So this is what the three stages of cell signaling is. Once the signal reaches to the cell, once it reaches it takes in that specific signal, and there is relay in the signal transduction itself. This specific pathway is called a signal transduction pathway. And once it reaches to the target wherever it wants to reach in terms of one specific organelle of the cell, it gives a certain cellular response accordingly. That's how a signaling happens. So first is reception of the signal by the target cell, then there is transduction of the signal within the cell, and then there is a response generated by the cell itself. So this was the summary of the fourth lecture. Then after this, sir, there is one lecture of one of the one of the doctor, which sir talked about neurosurgery itself, wherein, just a minute, I'll just read this in a time. So I was talking about the lecture 5. So this is what the sir generally talked about in his initial 4 lectures. The initial 2 lectures about why biology for engineers, and little fundamentals of biology, how exactly cell looks, what are the certain components of the cell, and how cell communication happens. That's what sir talked about in his initial 4 lectures. And in the fifth lecture, clinical perspective was given how exactly engineers have helped the biologists in order to cure one of the disease, like for example brain tumor, which sir talked about in his fifth lecture. You all can go through that specific lecture as well, wherein one of the doctor from, I think, Tata Hospital or Attract, I missed it, but that specific doctor talked about how neurosurgeons do the surgery by removing the brain tumor. So as a biologist, we know how brain looks, what are certain components uh, of the brain which are very much essential, and if there are certain components which can be ignored as well, like if even if they are not there, it is not drastically affecting. But there are certain parts of the brain, like for example, which controls the motor skills. For example, like if I'm trying to move my hand like this, it is moving, right? Because my motor skills inside the brain are proper. But if someone tries to change that specific part of the brain, then I might not able to move my specific hand because there is a damage in my motor skill area. So they need to be cautious enough when they are trying to treat trying to remove a tumor inside the brain, and they might make sure that they are not touching those specific parts of the brain, and they are reaching the tumor by some other direction and trying to remove the tumor. Now, in order to visualize the tumor, there are certain MRI scans available. Who made those MRI scans machines? Engineers are the one who made it, which are actually helping as a biologist or doctors who study biology, etc. Like they can visualize the brain in much more better manner, and they can detect that this is the location wherein tumor might be present. Similarly, in terms of while treating the brain, like we are not exposed to the whole brain while treating, right? Only a part of the brain is exposed. And that's how, like for example, if there is a tumor this side, only this part of the brain will be cut and this will be exposed. We are not exposed with the whole brain. So identifying where exactly you're trying to go inside the brain, it is very difficult for a doctor to see. So there are certain cameras which are being designed, or certain chemical reactions which have been designed so that with the help of that a tumor will glow in a different color and normal tissue will be seen in a different color, whatever. So all these concepts are merged together in order to make one successful treatment, like or maybe certain robotics machines are now being generated. Like all those are possible only if biologists and engineers come together, and be like those two people have already come together, and that's how certain diseases are now currently a bit easier to treat as compared to that of if there was no integration between these two topics, then biologists will be very difficult for them to treat any specific disease. Similarly, for engineers, they would not be able to solve any biological problem as such if they might have not built certain machines which are relevant to biology, or they would be only able to build those only if biologists give certain parameters for them, so that we basically want like this or something like this, which will be able like which will enable us to treat certain diseases, etc. So that's the reason those integration is very much essential, and that was the main aim of this specific course, basically, like integrating engineers and biologists together for so that we'll be able to solve a number of different problems, may it be related to biology or not. But if there is something in relation with biology, how exactly these two people can come together and try to solve different issues, basically. Hope you all were getting it, like whatever I tried to say. If you all had any specific doubt related to any of the slide, etc., I can explain you in detail about those specific topics as well, like if you all want that. First is interface with biology, second is medicine, biosignal processing from a medical electronics background. At my level, which course do you suggest will be more beneficial for my foundation and future core career? Like which course in terms of which are available in NPTEL or in general? The current course which you have taken, like it is very much relevant for you, I think so. Bioengineering one, and in general for medical electronics. So basically, I am a PhD student at IIT Bombay from Department of Chemical Engineering, though, but my whole study is in biology. Like in terms of my BSc is in life science, my MSc is in biological science. So I can give you better subject suggestion related to biology, but in terms of electronic stuffs, even I'm not that aware exactly related to biology or related to medical stuffs. I can tell you about the courses which are better, but in terms of electronics, even I am not aware that much to tell you exactly which course might be beneficial for you basically. Still, I can go through certain courses if I feel like that specific course might be beneficial for you. Then I can accordingly write it in maybe in the next lecture or coming few lectures. Yes. So till now everything is good. Everyone, are you all getting it? Whatever I tried to say. Although this was very basic lecture, initial one, like most of you might have understood because you have already went through the this specific basic topics way earlier in your earlier classes. If you're currently in your BSc or BTech, so in 11th and 12th only, or else during your school days, you might have went through all these courses, like all this part of the course itself. So this might be like a revision for you all. So maybe we'll take a break of 5 to 10 minutes, and we'll go through question and answer session. I have few questions of previous semester, so I want you all to interact during that specific question and answer session. Like I'll just display the question. I want you all to answer it in the chat box, or if you want, you can unmute yourself and then answer whichever way possible for you all. We'll start at around maybe 7:10 or 7:50. We can take a break of 10 minutes or so, and then we can start. Thank you. Are you all there? Are you all back? Give me a thumbs up, everyone. I'll start at 7:15 though, but we can just stop maybe for 5 minutes. Where are you all from? Like you all are doing your BTech from which college or which institute? Where are you all originally from? Like I'm originally from Mumbai itself, and I'm doing my PhD at IIT Bombay itself. Yes, you all can write it in the chat box, or if you want, you can unmute yourself and answer too. Yes, everyone. You are not back right now. I think yes, Ananya, Anu, Ashina, Ashlesha, Darpan, Arvai, Kanmani, Ishbi, Anushri, someone called as P, I don't know, Rimjim, Siddhi, Somadeep, Supershri, Veer Raheja, and Vijayashri. Yes, I just asked that where are you all from, like, or else it's not that, which you're currently from which college, like whatever degree you all are pursuing, is from which institute or which college. Somadeep says that he's originally from West Bengal and is currently doing from SRM Chennai. Great. So you are in your first year, second year. So BTech in biotechnology itself, if I'm not wrong. BTech first sem. Okay. Most of you all are from SRM itself. I think two of three people replied and they are from SRM itself. One is from Amity University, Noida. Great. Darpan. Fourth sem. What is your name? You are just writing your initial P. Pranav. Pranav. Nice. Nice name. Ashlesha is from Maharashtra, Nagpur, pursuing MSc Biotech in RTMNU. Okay. Central University of Jammu, first year. Pushpi is from UP, pursuing BTech at Sudhani University, Himachal Pradesh. Nice. This is nice. Okay, most of you all are in your first year itself. I think only few of them, like few is in MSc Biotech too. But So for BSc first year, I think this is a great course for you all. MSc also it is a good enough course. Like later it becomes a bit complicated in the last few lectures, but initial lectures it is good for like BSc first years also. Like you all will be learning different concepts related which so this is a bit of more integrated kind of a course, not related to one specific uh, topic or course, which is generally done in terms of any courses in your colleges or institutes. Like you might learn the same topics in two or three courses instead of learning it in one course itself, like part by part. I'm not saying that three course ka portion isme hai. I'm just saying that like few topics are from one course, few topics are from another course, and few are from another, like, which could relate bioengineering basically. That's one question from Shuloni itself. Where can I find syllabus for this course? You don't need any syllabus as such. Whatever, like the lectures are long enough. Like sir has taken 4, 4, 5, 5 lectures. But if you want syllabus, like there are certain references which sir has given you all. Like you all can download that specific book. Like I'll just show you. See, this is one of the reference Campbell. So you all can download this specific book. Like there are ebooks available for this specific book. You all can go through this specific book as well for going through the initial topics, whatever has been taught in the first few lectures. I don't know if I have shared the screen. I was showing the link of so few of you all are from Himachal, few of you all are from UP, some from West Bengal. People are learning at Chennai. So a diverse crowd here, good enough crowd. So I was showing you this specific page basically. So there is a book called as Campbell Biology. You all can go through this specific book. There are ebooks available for this book. You all can download that and you all can read the material present in this book, which is related to the course which has been taught here. Yes, so everyone is here. Can we start with the Q&A session? Yes, everyone please. I could see only Rim Jim gave me a thumbs up. How about others? Yes. How about others? Can we start? Or do you all want to talk more a bit? Okay. So what I'll do is again share the screen so that I can have this chat box beside while where's the chat box here. Okay. Yeah, this will be good for me. Okay, so here is the first question. So which of the following is not an example of biomimicry in engineering? So basically you need to compare uh, biological nature and assume that that biological nature was actually used for making those engineering stuff, maybe Velcro, wind turbines, solar panels, nanotechnology, whatever. So naturally, they have looked for something in the nature, and that has inspired them for making the engineered products, which are Velcro, wind turbines, solar panels, or nanotechnology. So, what do you think? Which one is not an example of biomimicry? Like I just explained what is biomimicry basically, and you just need to think of an answer which is not an example of biomimicry. Which one do you all feel? Write it in the chat box or unmute yourself and answer. Is it Velcro inspired by birds or wind turbines modeled after seashells, solar panels imitating plant photosynthesis, or nanotechnology in medicine? Okay, Ananya says D, and the answer is D itself. I want everyone to answer so that it will be a bit interesting. Yes, Ashina, you're right. So instead of writing the whole sentence, maybe you all can. The first option is A, second option is B, third option is C, fourth option is D. So that it will be easier for you all to type as well and answer it much more quickly. So how can biomimicry in engineering contribute to sustainability? I hope you all understand what sustainability is. So Khushbu says C, by promoting innovative and eco-friendly solution. Ashlesha says D, by ignoring the principles of biology. Who is it? So the D answer was for the first question, Ashlesha. Okay. So the answer is C. Yes, you all are right. The answer is C. By promoting innovative and eco-friendly solution. Yeah, even sorry to interrupt, sir. Can we get these questions after the session if possible? You will be getting the whole PPT itself along with the questions. Even the recording of this lecture. Like if you all want to go through the lecture once again, once more, you all can go through the lecture by looking at the recording of this lecture. So I think you all will be provided with a link wherein all these specific notes, like the PPT of it or the PDF of this specific file, is uploaded so that you all can go through that link and download those specific PPTs. Similarly, you all be given YouTube link as well, wherein so basically I'll be uploading this recorded lecture in YouTube, and you all can go through the like if you all want to revise the specific lecture once again and go through the lecture once again, you all can visit that specific link and watch the lecture, the recording lecture, basically, the recorded one. So you all say it's A, budding east, one is budding east, second is fission east, and third is ascomycete. What's the YouTube channel name? It's my name itself, like Gaurav Ganesh Bingi, but the link will be provided for you all so that it is easier for you all to find. Yes, the answer is one A itself. One is budding east, second is fission east, and third is ascomycete. So mitochondria is this is the fourth question. Mitochondria is referred as powerhouse of the cell. What biomolecule is mostly produced and used in the inner mitochondrial membrane of animal cells by oxidative phosphorylate proteins and produce ATP. So ATP is produced, but after oxidative phosphorylation, But we do oxidative phosphorylation of what biomolecule inside the mitochondria? Ashima says D, Soumyadeep says C. Most of you all are saying C. You all are saying C, but please look into this as well. Inner mitochondrial membrane of animal cell and not plant cell. Yes, the answer is D. And if it was plant cell, the answer would be yes. The answer would be C if it was plant cell. But it is animal cell, so answer is NADH. So while eukaryotic cell and prokaryotic cells have several similarities, one important distinction in the eukaryotic cell have membrane-bound organelles. Which organelle among them is following involved in protein modification and drug detoxification? So you all are saying it's C. Yes, the answer is C, which is Golgi apparatus. Okay. The nucleus contains genetic material in the form of DNA that we know, and its integrity is essential for cell function. Yes. Which of the following components is responsible for regulating the passage of molecules in and out of the nucleus? So, how signaling happens with the nucleus and cytoplasm with the help of what? And that's what plays a role in genetic transcription and replication. Replication of DNA itself and transcription of DNA to RNA. So the communication is happening between the nucleus and cytoplasm. That is happening with the help of, you all say A and B, both A and B, nuclear pore complexes, nuclear envelope, nucleolus. Most of you all are saying D, but the answer is nuclear pore complex. That's the major one. Even nuclear envelope might have a slight role, but the one which is the pores which are present, they are the ones which are helping in and out of the molecules, the nuclear pore complexes. In the membrane itself, there are nuclear pore complexes, and those pore complexes are the one which are helping you all. Envelope is the membrane of the nucleus itself, right? That's the envelope. But in that envelope, there are pore complexes present. The question is, which is responsible for regulating molecules in and out of the nucleus? It's the pore complex which is responsible, right? Because there are pore present. So that's the answer. Then you just have to label this specific thing. What might be this, and this in terms of a bacteria. So people are saying it's blee. One is flagella, second is pili, third is nucleus, fourth is vacuole. Actually, this diagram is a bit confusing. The answer which they have provided is mitochondria, though. Third one. So although this doesn't look like mitochondria here, it is nucleoid itself. Like I just read the B option basically. This there is something wrong in the. So this was the question which is given in the previous year assignment itself. Yes, there is no right option. uh, But I corrected that specific answer here in terms of while giving you answer for this question. So one is flagella, second is pili, third is nucleoid, and the fourth one is because it's a bacteria and not it is not a eukaryotic cell to call it as a nucleus. So it's not a true nucleus, so it's called as nucleoid. And fourth one, what they say is mitochondria, but it kind of looks like vacuole also. Uh, so we cannot exactly say that because in the article, whatever has been provided to me, they say that it's mitochondria, but it doesn't seem like mitochondria. It seems like out of all of these options, it's somewhat feels like vacuole, but not mitochondria though. And this is obviously not the nucleus. So this cannot be the answer because it's not nucleus, it is a nucleoid. So only one option was present with nucleoid. So they gave this answer as C. Like it should be C, but even first option is not pila, it is flagella. So there was something mistake, there's something wrong in this options as well. So I corrected these options by keeping it as a mitochondria as the fourth option, but it can be vacuole also. It is there's some mistake in the options itself. The eighth question is phospholipid bilayer makes up the majority of the cell membrane as we just saw in terms of gram positive and gram negative. So although they also include in integral and peripheral membrane proteins, which of the following functions as transporter, receptor or enzyme for various cellular processes? The internal one, the peripheral one, or some other proteins. Ashima says B. Yes, the answer is internal membrane, integral membrane proteins. Okay, haploid gametes are produced during meiosis in the setting of cell division. Which unique events result in genetic diversity in progeny during meiosis 1 and not during meiosis 2? Yes, you are right. Mitochondria is responsible for energy production and is believed to be originated from ancient symbiotic relationship. In this theory of endosymbiotic theory, which two are thought to have contributed to the evolution of mitochondria? Yes, it's alpha probacteria and archaea. What is biomimicry in engineering? So this was one of the assignment. This was some other assignment also, previous years. So what is biomimicry engineering? We did discuss about what is biomimicry. It's imitation of natural designs and processes in engineering. Yes. Which of the following statement is are incorrect about viruses? Yes, it's B. Plastids and mitochondria are two important organelles of a plant cell. Which of the following features are exhibited jointly by both of them? Plastids and mitochondria. It's A, B, and C. They both have prokaryotic origin, they both have double-stranded DNA, and they both have 70S ribosomes. Yes. So you just need to look at this image and what is the function of the plasma membrane specialization shown in the image. To allow direct diffusion of ions and small molecules between adjacent cells, to help encourage of centromeres during cell division, cause cytolysis and to provide attachment to each other during severe stress. It is D. You all initially said allow direct diffusion, so there is nothing like communication happening. It's the part of a skeleton which helps you all to provide attachment, and not for this there are gap junctions present. So this is not a gap junction here. Okay, so there is Ramesh who is attending a biology class. His teacher asked to observe onion cells. In your initial 11th and 12th class, you might have observed onion cells under a microscope, right? So you observed a cell in two different nuclei and a thin cell plate dividing the two chambers. So this specific line will tell you the answer itself. What stage of mitosis did Ramesh observe? Cytokinesis, because thin plate is being already being seen. So it's cytokinesis. So which of the following characteristics distinguishes eukaryotes from prokaryotes? It is efficiency of processes here. Okay. Improved cell mobility, streamlined translation, presence of cytoskeleton, or compartmentalization of cellular process. Yes, you all are right. The answer is D. Just a minute. Okay, we are done. These were the questions which I had. So we still have some time. If you all want to ask any questions, we all can discuss related to that. So mitosis is divided into two different phases basically, or what do we say? Not even three. Like there is G1 phase, there is S phase, there is G2 phase, there is M phase. So so you cannot say it is a part of mitosis, but it is a part of cell division. Because M phase is the one which we call it as mitosis phase, right? So in M phase there are four phases itself: prophase, anaphase, metaphase, telophase. After that there is a step called cytokinesis. Paracrine signaling and its significance. So paracrine is the one which is being the signaling wherein the signal happens between adjacent cells, and that so if something is happening in any of the corner, the signaling happens from cell to cell. Like for example, what example should I give? Immune cells, immunity cells, uh, the one which is responsible for our immune system. So the signal needs to go to in the whole body where that this specific location needs certain macrophages or phagocytes, etc. So that can only be possible after a paracrine signaling. So there are certain inflammatory products which will be released at that specific location where immune cells are required. Those specific inflammatory products are nothing but the signaling factors, and that will go through each and every cell and try to tell the whole body that this is the location where we require more amount of immunity cells, so that like macrophages or phagocytes, etc., in order to treat that specific inflammation area. That part of signaling can be called as paracrine signaling. Yes, anyone else? So that's what I'll be doing basically in like in the next lecture also on next Tuesday. So whatever sir might have taught in his 5 lectures, I'll give a recap of all of those 5 lectures, and there will be certain question and answer sessions. So we'll go through those question and answer so that it will help you all to solve assignment. And in this class, basically, you all can ask questions related to your assignment also, like while solving the assignment after submission, basically. And but still, while solving it, I can give you all hints to solve the question, but I cannot tell you the answer exactly. The answer needs to be found by yourself, and you all need to submit the assignment. But in order to solve that specific assignment, I can help you all to give certain hints or try to explain what the question is asking about, etc. So that it will be easier for you all to answer that question. And after the submission of assignment, if you still have any doubts related to any of the question, we can discuss that in the class. Similarly, when you go through all those 5 lectures of the of that specific week taught by sir, after going through those lectures, if you all have any kind of doubts related to that specific those 5 lectures of that specific week, we can still discuss on that as well. Ethylene signal transduction pathway. It was mentioned briefly in the 4th lecture. If you want that specific pathway to be discussed in detail, I can take in that specific question and explain you all in the next lecture. So whatever I remember related to ethylene is, I think ethylene is used for fruit ripening, something like that, in terms of a plant. So if ethylene is being induced in a specific plant, and the fruit ripening basically happens, enhances fruit ripening. If I'm not wrong, I'm not sure about it, but I think that's what the function of ethylene it is. But if you want that pathway to be explained a bit more in detail, I can talk about it in the next lecture. Yes, anything else you all want to ask me, or do you all want me to change the way I am presenting? Anything related so that it is easier for you all to understand, or I should go a little slower or a little faster, or should I incorporate few more questions, or like these many questions are enough, or whatever, like n number of suggestions, whatever suggestions you all want to give in order to make your understanding better, we can go through that. So you all want more questions to be there? Okay, I'll try to include more questions. But I don't like the problem with adding more. I will try to add that, no problem, no issues at all. But for in terms of few topics ahead, like in terms of courses, I might take more time. Like today, I took only like an hour or so, maybe like 1 hour or 1 hour 10 minutes in order to explain 4 lectures, like in brief, in much more quicker manner. Similarly, for next week lecture, week 2 or week 3, they are a bit more into biology. Like as you all are from biotech field, I think it would be all for easier for you all. But in terms of Luther, which is from computer science background, he might need a bit more detailed explanation. So depends. Like I'll try to finish it as early as possible, and similarly, I'll try to include more questions. But I think yes, I can take that and I'll try to include more questions if possible. Yeah. And like even it is fine even if you all don't call me sir, you can call me my name as well. I'm still a PhD student, although I'm teaching you all in this course, and I have been teaching since many years. So like after my MSc itself, I have started my teaching career. Although I'm doing my PhD right now, but there's no need of calling me sir as such. What inform the pattern of exam paper, like how many questions and does it go out of the syllabus? That I need to confirm with NPTEL people once, and I can let you know that. But mostly it is on the base, like out of syllabus in the sense it will not be completely out of the syllabus, but related to the portion which is being taught. It will not be like straightforward questions. It will be related to the topics which is being taught. So you all need to know a little more background. So whatever topic is being taught, along with that, a bit background related to that topic also you all need to know, so that it will be easier for you all to answer the questions. It will not be like the like whatever you might have studied during your school days or initial 11 12, like ये एक क्वेश्चन होगा और उसका आंसर टेक्स्ट बुक में होगा ही. It will not be exactly similar to the way you might have taught in your earlier classes. This will be a little not exactly straightforward, I meant to say. Scope of biotechnology, how it will impact the future. See, biotech is something which will never fade. Basically, it has a good feature. Like so, basically, I am doing biology or I am doing sciences just because I love doing sciences. The other thing is, if you are doing it in India, inside India, you might not earn that much, but just because you love doing sciences, you will love your job no matter how much you are paid. But if you want to earn, maybe this is not a good field in terms of India's payment in terms of this specific field, but want to earn well in the same field, maybe outside India, they pay much better for doing the same amount of work or same skill of work which biotech people do. In terms of impact in the future, there is a huge impact of biology, and currently, if you see the number of diseases being increasing, and so basically, even so, doctors are the one who treat the diseases, but uh, in order to make a successful drug or a successful uh, antibiotic or whatever you mean to say, all those tests are done by researchers itself. So if you are in a field called as BTech in biotechnology, so you'll do your BTech, you might do your MTech, and if you in that during your BTech and MTech itself, or if you want to pursue PhD ahead, whatever, in that specific period itself, you all are having internships or something like that where you will learn how exactly research is being done. So that research itself is currently very important, so that you will end up having a great technique or a great drug or whatever, so that it treats the disease. So if you all see, currently antibiotics are increasing, like basically bacteria are building their own mechanism in order to resist each and every antibiotic. So there is, in coming future, I'll say there will be a need of finding out a new antibiotic itself. And if you all, do you all know the story of penicillin, how exactly penicillin came into picture? Yes, no. Yeah. So during that time, whatever penicillin came into picture, so at that time, bacteria were not that resistant, and they were not aware of those specific antibiotics, and we were able to kill them very easily. But currently, due to use of like overuse of certain tablets or misuse of certain tablets, bacteria have developed n number of resistance. And in coming future, there will be a tremendous need of finding out new antibiotics. Like that is one of the part of the whole research. There is n number of other topics also. Like it just reminded me of an antibiotics. I'm just relating it with your question, but that can be one huge impact which is actually needed in the future. But similarly, there will be a number of other stuffs as well. So there is a good scope. It's not that Battlefield has doesn't have any scope, but if your aim is to, as I said, if you want to earn more, then you need to go out for earning more. Like here, still you can earn, you can do good thing in India also, but you'll not be paid that much maybe. Which is better, academia or industry roles in biotechnology? Academia or industry, and also which countries are best for masters in biotech? Okay, so in terms of academia or industry, uh, both are very different in nature. In terms of academia, you will be teaching and doing your research. In terms of industry, you will be assigned a specific role, whatever role is assigned to you, and you will be going through that specific. For example, you are managing certain projects, so you will be involved in different number of projects, and you will be just taking updates from each and every project. If you are a part of one of the project or doing R&D, so in that R&D also you will be a part of one project itself, like related to what all research you need to do for that specific product development or whatever. That's what industry roles are. So both are very different, and whichever is of interest to you in terms of academia or interest, you should go for that, and do not look for how much you are earning in academia or industry. Just do what you love and what you are more interested in. That's what I think your answer for academia or industry. And which countries are best for masters in biotech? uh, as far as one of few of my friends have been in terms of doing masters in biotech, one of my friend is in New Zealand, which she does masters in genetic engineering over there. I think that is one good country for doing masters in biotech. But in terms of having a good biological, like, what do I say, having a good biological context or contact which you can build in region, it would be USA most probably, and UK as well. Like they are having in terms of biotech field, UK and Germany is good, but in terms of Germany, the engineering perspectives are much better as compared to that of biotech, maybe like automobiles and stuff. Those are much advanced in Germany as compared to that of biotech. Like I'm just comparing. Even all other foreign countries are good in biology field. I'm not saying any of the countries not that great, but the one which are bit better than other countries, I would say US and UK and New Zealand as well. That's my opinion. It's not that that is for sure. It's just my opinion that they are little better. Also, all other countries are equally good enough in terms of batting. Even India is a very good field, like having a good scope in biology, but just that payment is a bit lesser as compared to other fields. Is genetic engineering a good field in biotech? Yes, it is a very good field in biotech. Yes, anything else you all want to ask? We have 5 to 7 minutes more. Yes. Okay, I think we'll be having our next lecture on today is 27, then I think first, 3rd Feb, right? Yeah, on 3rd Feb, we'll meet for week 2 lectures. So I'll go through those uh, 5 lectures which Sir has talked in week 2. I'll go through those with initial 1.5 hour, so that it becomes a revision for you all. Then we can discuss few question and answers. And similarly, as I said, if you all have any kind of doubts related to initial lecture or while solving your assignments, you can talk about that questions as well. Okay, if that's it, I think we can end the lecture. If you all have any questions, still we have 5 minutes or so, or else we can end the lecture. Okay, then thank you. Thank you for attending. Thank you for joining. We will meet on next Tuesday, that is 3rd Feb. Thanks.